Hello, everyone. Good afternoon to all the doctors who have joined us from the Middle East countries, and good evening to all the doctors from rest of the Asia Pacific region. Today we'll be talking about Embrace. So we have named it Embrace. Actually, it is non-invasive PGT-A. And we have none other than Dr. Carmen Rubio herself, who will be speaking on the topic. Hello, Dr. Carmen. Dr.
Hello.
Carmen, hi, can you hear me, Dr. Carmen? This is Vikram from India.
Yes, hello. Good
Hi,
afternoon.
welcome.
Thank
Very,
you.
very good afternoon to you. Welcome to this episode of our Igenomix webinar series, which we are exclusively holding for the audience of Middle East and Asia Pacific. I welcome you on behalf of Indian team, on behalf of Middle East team, on behalf of Japanese team, and on behalf of the audience. It's a pleasure seeing you here with us today. So, Dr.
My
Carmen, before I hand over the session to you, please permit me a minute. I would like to introduce you to our August audience, which is here with us today. So, Dr. Carmen Rubio, I think this name definitely rings some bells in your ears. She's a well-known name when we talk about reproductive genetics. And let me brief you about Dr. Carmen's experience. She's profoundly experienced, but I would like to, name some of her experiences here, read them out for you. Dr. Carmen is trained in science in the University of Valencia, Spain. Dr. Carmen Rubio specialized in cytogenetic studies in human reproduction, partly in the University of Barcelona. Dr. Carmen completed her PhD in the field of reproductive genetics and postdoctoral research, including the research in male and female meiosis at the laboratory of Dr. Patricia Hunt and Terry Hesselt, which was in Washington State University, USA. As you know, she has published, you must have read a lot of her publications. She has published more than 100 papers in the main peer-reviewed specialist journals in the field, book chapters, as well as numerous lectures at conferences worldwide. Currently, she is the head of research for embryonic chromosomal abnormalities at Igenomix, focused on non-invasive approaches of embryo and Ploidy. Furthermore, Dr. Carmen has 218 publications to her credit, which have received 22,970 reads with more than 4,300 citations. So it's undoubtedly a pleasure, Dr. Carmen, to have you here with us. The session is all yours.
Thank you very much. It's my pleasure to be here today and to share our experience at Igenomix in this exciting field of the analysis of the embryonic cell-free DNA secreted to the culture media during embryo preimplantation development. We know that chromosomal abnormalities are really very important in human reproduction because chromosomal abnormalities can cause miscarriages and can cause implantation failure. In the last 25 years, there has been several efforts to try to identify the presence of chromosomal abnormalities in human embryos, and the most extending approach now is the Trophectomy Biopsy, which implies an invasive technique with the removal of 5 to 8 trophectoderm cells from day 5 or day 6 blastocysts, and then we can just place these cells in a PCR tube and we can analyze with the current technology that is NGS technology. With this technology, we can identify abnormalities for the chromosome number and also for chromosome fragments. You can see here an example with a monosomy for chromosome 16. But as in other fields in medicine, we have been searching for non-invasive approaches to be able to identify chromosomal abnormalities in human embryos. And this is the latest research that has been done by our team. So our idea was to see if the embryos, when they are in culture, before transfer the embryos to the uterus, they secrete DNA to the environment closer to them, to the sperm culture media, with the same idea that in the non-invasive prenatal testing, in which the fetus is secreting fragments of DNA from the placenta to the blood of the mother. So we were trying to see if this is also happening before implantation, and we learned that the embryos, in fact, they start to secrete small fragments of DNA to the culture media. That means that we can analyze the culture media in which the embryos has been growing during the IVF treatment, and we can do sequencing of this media, and we can have also these nice results in which we can identify the chromosomal content of the embryo. So this is the summary of the research and the status today of these approaches for chromosomal analysis of embryos. And we have learned in the last 5 years that the embryos start to secrete these small fragments of DNA from the very beginning of their development, when they start to divide and increase the number of cells. But this phenomenon is really enhanced at the morula stage. On day 4 of development, we can start to see higher concentrations of these DNA fragments in the culture media, and the concentration increases as the number of cells in the blastocyst increases. We have defined that the right moment to collect the media to have the most robust results is on day 6 of embryo development, and the special moments in which we need to place the embryos in special culture conditions are from day 4 to day 6. During these 2 days, we can have the embryos in a small drop of culture media, and then later on we can analyze this drop and we can just sequence it and identify the chromosome number of the embryos. And this technology is not new, in the sense that the research started more than 5 years ago, and the first publications appeared in 2016. The first publication by Samunki and collaborators was a proof of concept, showing that in fact the embryos were releasing DNA to the culture media. And this was very new, because nobody expected at that time that the embryos can secrete DNA to the culture media. Later on, there has been several attempts to see the consistency of these results, and the design of most of the studies published so far consists in the comparison of the data obtained from the trophectomy biopsy with the results of sequencing the culture media. So you can see how the concordance rate between these two biological samples from the same blastocyst has been improving in the different studies. We can have concordance rates of 85%, 87%. So the results are encouraging in the sense that higher concordances are achieved by different groups. So this slide represents the summary of the publication so far. And I would like to point out in this slide that in all these studies published so far, the authors in some way were manipulating the embryos before collecting the media. In many of these studies, day 3 assisted hatching was performed, so the embryo had a hole in the zona before collecting the media. That can enhance in some way the communication and the release of the DNA. Other embryos, in other cases, also vitrification was included before. For example, in this study with these very good results by Xu and collaborators, the authors performed day 3 assisted hatching, then they thaw the embryos on day 3, and they keep on culture until blastocyst stage. So day 3 assisted hatching and day 3 vitrification were incorporated. We cannot consider this as non-invasive or fully non-invasive, and nowadays there is consensus on considering this as minimally invasive, as well as the other procedure that some authors have proposed, that is to collapse the blastocyst with some laser shots to secrete the blastocellular fluid to the culture media and to analyze both together. So we can consider these approaches as minimally invasive, but not fully non-invasive. And you can see that with these approaches also, concordance rates in 3 studies were compared with the full blastocyst, not just the trophectomy, and the results are very encouraging. So with this idea in mind, we wanted to develop an approach to be able to achieve this high concordance rate, but without previous manipulation of the embryos, without previous hatching, previous vitrification. So we started with our research, and the first publication appeared in 2018 with a proof of concept. in human reproduction. In 2019, we published in Fertility and Sterility our pilot study with a very well-defined protocol for the IVF lab and NGS, and also we were able to assess the clinical impact of the abnormalities found in the culture media. And just recently, last month, we published in the American Journal of Obstetrics and Gynecology a multi-center prospective study in which we were able to test for consistency of these results among different centers. So let's go on with the first study. In this first study that we published in human reproduction, we keep the conditions in the IVF. We didn't include any modification in the regular protocol of the IVF center in which we were performing this study. So the IVF center was using embryoscope. They were PGT-A cases in which the media was changed on day 3, and then the embryos were cultured in the embryoscope until day 5. The volume was the regular volume of the embryoscope, 25, 28 microliters. We were able to collect about 20 microliters from the wells, and we were doing the regular and our conventional protocol of NGS for regular cases of trophectomy. And what we observed in this study, comparing the trophectomy biopsies with the media, is that the concordance rate was 30%. So it was not nice. It was much better than the first pilot study, 30%, but 30% was very low. And we were really disappointed because we observed partial maternal contamination or full maternal contamination. In total, we observed contamination in about 60% of the samples. Most of this contamination come from the cumulus cells that surround the oocytes. So we have to get very careful with this source of contamination. Also, we observe contamination from the polar bodies. DNA from the polar bodies can mask the result at this level on day 5. And we observe also some external contamination from the environment, maybe from the embryologists, from the surfaces in which our dishes were in contact with. So we learn a lot with this study. And we say, okay, there is something there. We were sure that there were DNA from the embryos because we were using SNPs to check for the origin of this DNA, but we have this issue with contamination. So we developed a project in which we wanted to improve the IVF conditions to minimize the risk of contamination, and we improved our NGS protocol to increase the detection rate. We wanted to estimate also the optimal time to collect the media during embryo development, and we wanted to assess the impact of the abnormalities observed in the media in the clinical results of the couple. So we published these results in Fertility and Sterility. This study was performed in collaboration with Genera Lab, with Dr. Filippo Baldi, Laura Rienti, and the main changes we did in the IVF protocol were the day in which we were changing the media, refreshing the media. We just delayed from day 3 to day 4. We were changing the media on day 4. And before placing the embryos in a new drop of media, we add a serial washes. That means we were washing individually each embryo by several drops to dilute and to minimize the carryover of DNA or material from the previous days of development. So we wanted to have the embryo very clean without any trace of DNA from previous day in the new culture drop. And the new culture drop was smaller, much smaller in size than the volume that we had in the previous study in embryoscope. We wanted to culture the embryos in 10 microliter drops to really to have everything very controlled and all the DNA secreted by the embryo concentrated in a small volume for a better NGS analysis. And the embryos were growing until day 5 or day 6, and the biopsy was performed at this stage. No previous assisted hatching was performed in these embryos. Assisted hatching was performed just at the moment of biopsy. That means that the embryos were being growing in the culture drop without any manipulation from day 4 to day 5, if the embryos were at the moment for the biopsy on day 5, or from day 4 to day 6, for those embryos in which the IVF center decided to do the biopsy according to the development. So at the moment of biopsy, the embryos were transferred to the biopsy dish, and the media was collected, placed in this PCR tube, and sent to us to Igenomix for chromosomal analysis. Regarding the chromosomal analysis, we incorporate several modifications from the standard protocol that we used until that moment for a trophectomy biopsy, and we were modifying particularly the whole genome amplification part. So we needed to amplify with higher efficiency the DNA from the biological sample to be able to apply the NGS. Here you have an example that if we were using the regular PGT-A protocol, in this agarose gel, you can see the representation of the concentration of the DNA. If the bands of the agarose are very pale, that means there are few DNA fragments, and if the bands are very bright, this represents higher concentration of DNA. That you can see that with the modified protocol is very robust in different samples. So those were the main differences from our previous publication, and then we test for the concordance rate between the trophectomy biopsies and the media in these 108 blastoses. 27 of them were day 5 blastoses and day 5 media, and the concordance rate by modifying these conditions was double that in our previous study. So we were able to double the concordance rate to 63%. This was nice to see. It means that our modifications were making an effect, but still 63% is too low, just thinking for a clinical translation of this test. However, when we check the results on day 6, the embryos were in this small culture drop for 2 days. We had 81 embryos that were biopsied on day 6. In this center, they do the biopsy on day 6 on most of the blastoses. And here you can see how the concordance rates increases to 84%. And this was very good number because this concordance rate was similar, as if you can remember from the other publications, in which they had manipulated the embryos previously. So we didn't manipulate the embryos here, and we can have very good and similar concordance rate, and the informativity rate was very high. So this allows us to establish the optimal time for media collection, and we established it to be early in the morning on day 6. And if we pay attention only to the day 6 media collections, you can see how the concordance rate was 84%. We also have an additional 5% of embryos in which the autosomes were concordant, but there was a gender discrepancy. And this is a very important numbers. We tested for the false negative and the false positive rate. The false negative were 2.5, meaning that in 2.5 of the cases, the biopsy was abnormal, but the media was normal. And the origin of this false negative is mainly because of the cumulus cells. So we know that we have to take care of the presence of DNA from the cumulus cells. That's why we add these extra washes steps just to dilute the presence at the end of the development. And also we have false positive in this study about 8%, 8.5%. And we don't like to call this false positive anymore because as you will see in next slides, we have learned that this is just discrepancies between the biopsy and the media. Because if we have a euploid biopsy and we have an aneuploid media, we know that the media, if is contaminated, is going to be contaminated by normal DNA, not by abnormal DNA, because cumulus cells had normal karyotype, and if there is contamination from the environment, from the embryologies, they have normal karyotype. So these aneuploidies that we observe in the media, we think they are very real, and they are the discrepancies just represent the presence of mosaicism in human embryos. And this was reinforced by the clinical analysis that we did in this study. In 29 patients, the IVF center provided us the clinical outcome after the transfer of embryos according to the trophectomy biopsy. In all cases, the IVF center transferred the embryos according to the trophectomy biopsy results. All the cases were euploid because the center didn't know the result of the media. We had the results, Igenomix of the media, but they didn't know. And they were providing us the results of the clinical outcomes after the transfer. So in 29 single embryo transfer, we saw that 17 samples had euploid trophectomy biopsies and euploid media. And then the results in clinical pregnancy rates were very good, 52.9, and no miscarriages were observed in this small subset of samples with very high ongoing implantation rate. However, on the contrary, when we had abnormal sperm-blastocyst media, clinical pregnancy rates were much lower, 33.3. Half of the patients had a clinical miscarriage, and the ongoing implantation rate decreases to 16.7. So this was giving us an idea that the media is giving us accurate information about the chromosomal status of the embryo, and also was giving us the idea that maybe with the analysis of the media, we can decrease miscarriages and we can improve ongoing implantation rates in general in every IVF patient. Based on these studies, we decided to go through a biggest study, including several centers, to see if the results can be reproduced among different IVF conditions, different culture media, different incubators. And this was the aim of this study recently published. So we designed this study with the idea to collect more than 3,000 samples distributed in 8 centers, and this publication collects the data from the interim analysis that we performed at the end of 2019. And as I mentioned, a total of 1,301 media and the corresponding trophectomy biopsies were included in this study. And in addition, we had the possibility of doing the analysis of the inner cell mass and 81 blastocysts donated for research from a USA center, Boston IVF. So in general, from the first part of the study, including the 1,301 embryos, the concordance rate between the culture media and the trophectomy biopsy was on average 78.2%, was a little lower than in our pilot study, but the good news is that in 4 of the centers, concordance rate were really very good, above 80%. You can see here 81, 86, 84, 85, 86. So you can really see how in many centers, concordance rate can be as high as 85%. And we think this is very reassuring for the clinical translation of this research. Also, we test for the impact of different conditions of the background and the patient, different infertility causes, hormonal profiles, different stimulation protocols and culture conditions. And we only observe differences in the maternal age. We observe higher concordance rate when the patient had higher age, but still in patients, young patients, concordance rate was very good. It was 75%. Regarding culture conditions, we didn't observe any difference among the 4 different media that were used in the IVF centers. Also, we didn't observe differences in the different incubators employed by these clinics. And in the second part of the study, as I mentioned, we were able to analyze the inner cell mass from 81 blastocysts, and we had informative results in 64 blastocysts, in which we have information for the 3 types of biological samples: sperm blastocyst media, trophectomy biopsy, and inner cell mass. You can see the different comparisons. All of them had very similar concordance rate. The media with the trophectomy 87.5, very good result. Most importantly, the media with the inner cell mass, 84.4%. So this result is showing us that the origin of this DNA in the culture media maybe is from both the inner cell mass and the trophectoderm, because the inner cell mass saw very good concordance rate too. And we have the comparison of the trophectoderm with the inner cell mass, and the result was 87.5%. Also, we did a deeper analysis, and we wanted to check the concordance rate for different types of abnormalities. So if we pay attention only to whole chromosome aneuploidies, that means the abnormalities that are from meiotic origin, from the oocyte or from the sperm. For this whole chromosome aneuploidies, that represents that the full chromosome is abnormal, the concordance rate were 92.2% of the media with the trophectoderm and 86% of the media with the inner cell mass. And trophectomy biopsies with inner cell mass had the very nice concordance rate of close to 94%. That is what we have regularly in our PGT-A program, because we know that if we think in segmental aneuploidies that are aneuploidies for a small fragment of the chromosome or mosaicism, then these different parts of abnormalities account for the differences between the trophectoderm and the inner cell mass. Another important message from this slide is that false positive rates were very similar between trophectomy biopsy and the media. But what is very important is that the false negative rate was very low in the trophectomy biopsy as expected. And we want to reinforce the idea that the gold standard technique for us, for a couple that wants to be very sure that if the embryo has a chromosomal abnormality or not, is the trophectomy biopsy. We still consider the trophectomy biopsy as the first approach for those couples that they don't want to go ahead with the risk of transferring an embryo with a chromosomal aneuploidy, because as you can see, the false negative rate is the best. In the sperm blastocyst media, we have a false negative rate of about 9%. And as I mentioned before, this is mainly due to the risk of contamination with cumulus cells. We know now how to get rid of these cumulus cells, so we need to work very closely to the IVF laboratories that wants to work with us to minimize this impact. But because we know there is the risk of this cumulus cell contamination, we can see the non-invasive approach as a prioritization system. That means that we can see the analysis of the culture media as an additional result of the embryo, additional information of the embryo. So we have the morphology, maybe we have the morphokinetics, and now we have the analysis of the media as another biomarker to help you in the IVF center to identify which embryo to transfer first. The idea is just most of the IVF centers all over the world are going in the direction of single embryo transfer. So if you have several blastocysts, you need to know which embryo to transfer first, because this embryo that you transfer first needs to have the higher probability of being euploid, because this will give you the best pregnancy outcome. Then you will have the lowest miscarriage rate. And also, if we transfer first the embryo with highest chances of being euploid, you will shorten the time to pregnancy, and then you will decrease the number of transfers needed to achieve an ongoing pregnancy. So this is the idea of the analysis of the culture media, and this is the idea of the embrace test, to give you and identify which embryos will have the higher probability of being euploid, and then you can decide which embryo to transfer first to improve the clinical outcome. But not just to say exactly if the embryo is normal or not. It's just a question of probability and having a priority order for the embryos for transfer. And with this idea on mind, we developed the embrace test, that is the embryo analysis of the culture environment to prioritize for transfer the embryos with a higher chance of being euploid. And because this is a non-invasive approach and is going to minimize the risk of transferring an abnormal embryo without the need of a biopsy, we consider that this could be a good approach for any IVF patient. So any patient, young patient, patient with male factor, any patient can benefit from this, because you can increase the probability of getting a pregnancy in the first transfer. So the benefits compared to a regular IVF cycle is just that we are increasing the probability of transferring a euploid embryo, resulting in a healthy pregnancy in shorter time with a lower number of transfers. But again, we have patients that had an indication for PGT-A, patients, for example, about 34, 40 years of age, that they can do the PGT-A with invasive biopsy, but in many cases, the clinicians do not feel comfortable with the biopsy, or even the couples, that they don't want the biopsy. We can offer the analysis of the media, also for couples with low number of embryos that they don't want to go through the biopsy. We can offer the analysis of the media, and the advantages for those couples with a PGT-A indication is that they can have a good information about the chromosomal status of the embryo without the biopsy, and also it will be a more cost-efficient treatment because the couple does not need to pay for the embryo biopsy, just for the genetic analysis of the media. So this is the frame of the technology and how we can foresee the application currently, because this test is now clinically available for you in all of our Igenomix labs all around the world. How we have developed this test, it's very important to have a consistent test and to understand how we should analyze the chromosomes in the culture media, because the DNA in the culture media is not as in the trophectomy biopsies, it's not DNA in cells, it's cell-free DNA, and there are smaller fragments. So the way in which we are diagnosing these abnormalities is completely different. So we have been working with our bioinformatician team to define the thresholds for Ploidy, That means to define when we consider that a chromosome is normal or not, and our threshold for ploidy in the media are different from the thresholds in trophectomy biopsies. We have identified different combinations of abnormalities in the media, and we have many different categories, and we have given to each category a different euploidy score, and the euploidy score represents the probability of this combination of abnormalities to correspond to a euploid trophectomy biopsy. So different combination of results in the media, we can give a euploidy score that is the percentage of blastocysts that even with this abnormality in the media can have normal results. And with this euploidy score, we have classified the embryos giving a higher priority to the ones that have higher euploidy score. And here you can see an example. This is an example of the report that we are going to release for the culture media. You can have the detailed information if you want for all the chromosomes. And also we are giving this graph with the euploidy score. In this example, we have 4 embryos. We have 2 media that have a normal result in the media. So these are going to be the ones with higher euploidy score that according to our concordance rate is going to be 81%. So if we have this result in a clinical case, we will transfer first the embryos classified as 1, but then we can have different combination of abnormalities, as I have mentioned. And for example, if we have a segmental duplication, in this example is a small duplication of a part of the chromosome 4, we know these chromosomal abnormalities are really benign. Also, we know they are mostly of a mitotic origin, and our euploidy score with the trophectomy biopsy has been 75%. That means that we can transfer this and we still have a very good chance of having a normal pregnancy. So we classify as second place for transfer. And then on the opposite side, we have other chromosomal abnormalities that imply the full length of the chromosome. For example, this trisomy for chromosome 17. When we have these abnormalities, we know that in most of the cases, in 80% of the cases, the blastocyst is going to be abnormal. Only about 20% of the cases, the blastocyst can be also can be normal. So this last category could be considered the last possibility for transfer. So this is the way in which we can advise couples about which embryo to transfer first, and we should also should take into consideration female age, because the potential advantages of using the analysis of the media will be varying according to the different female age. So if we have a patient, as an example, below 30 years of age, then the probability of transferring a euploid embryo, just according to morphology, is about 60%. So if we compare to the probability of transferring a euploid embryo using the embrace, is going to be increased from 60 to 80%. It's going to be a good opportunity, even for young patients. For patients, older patients, we know about 40 years of age, the probability of transferring a euploid embryo according to morphology is about 20–25%. So in those couples, if we use the embrace test, we increase the probability of transferring a euploid embryo from 20 to 80%. So this is a big advantage for those couples. So this is the way in which we can foresee the benefits of this technology applied to any patient in IVF. So to conclude, with this presentation, we can summarize what the main findings of our research in the last 5 years. And you can see how we have been able to identify the presence of embryonic cell-free DNA, and that we can amplify, we can sequence, and we can have a representative information of the embryo chromosomal content. Also, that the concordance rate between the trophectomy biopsies and the media can be as high as 84 or 85% if we collect the media on day 6, and these results without any previous intervention in the embryos. High concordance rates can be achieved also if we compare the results with the inner cell mass, and our euploidy score and our prioritization system will allow us to transfer embryos according to the aneuploidy risk, and will allow us to increase pregnancy rates in patients, decrease miscarriage rates without the need of an embryo biopsy. So I would like to end this presentation by thanking, first of all, Professor Carlos Simon the head of the scientific advisory board of Igenomix, and really the promoter of this study, because he encouraged us to look for non-invasive approaches for the analysis of the embryos. I would like to thank my very nice team working together in the lab. We are the research team in non-invasive strategies for PGT-A. Luis, Carmen, and Lucia, we have been working together in the last years just to present you today these results. I would like to thank also the clinical studies team at Igenomix, because they were really working hard to present all the projects in the local IRBs of the many different centers involved in these studies all around the world. And also, I would like to stress that they were monitoring on site in each IVF center for the accuracy and the consistency of the results. And finally, I would like to thank Professor Nicolas Placha, working in Singapore and now in the States, because he was supporting us to try to understand the origin of the DNA, working with live imaging system to identify if there are fragments, the dynamics of the DNA during embryo development. And thank you all for your attention.
Well, thank you, Dr. Carmen. I don't know I can thank you much over this. It was indeed a pleasure to have you here with us and taking up this session, sparing your time for this. And I'm sure the audience also thinks likewise. So thank you very much on behalf of Igenomix team and on behalf of the audience. I would also like to thank the audience for being so patient and, trying to understand the subject, which is definitely a bit novel for many of the doctors in the audience. Well, before we wrap up this session, Dr. Carmen, we have a lot of questions with us. And can I start reading out the questions to you, Dr. Carmen?
Yeah, sure, a pleasure.
Okay. This is Dr. Rita from Bangalore, India. She has a question. How is higher concordance explained with increasing age?
Okay, I think that this is because with increasing age, we have a higher percentage of embryos that are truly aneuploid. And if we have an embryo that is truly aneuploid, imagine that we have a trisomy for chromosome 18. Even if we have a small contamination, we can identify the trisomy. So if we have a very clear abnormality and if we have a small contamination, not too much contamination, we can identify the aneuploidy. So I think it's because of that, because with advanced maternal age, we have higher percentage of abnormal embryos, and the concordance rate is really very good in those cases.
Thank you very much, Dr. Carmen. Another question comes from Dr. Arveen. She's also from Bangalore, India. Is there any method for quantifying beta HCG levels in spent blastocyst media? If so, are you checking beta HCG and how do you check?
Okay, we are not checking beta HCG. There has been many attempts to check for hormones and to check for different metabolites in the culture media. And our experience, Dr. Carlos Simon had a very interesting project starting like 15 years ago, trying to identify different metabolites and different biomarkers in the culture media of human embryos. And what his team observed at that time is that the results were not very consistent. There were very differences based in the limitations of the detection limit in the technology. So the levels of these metabolites in the media are very low, and the sensitivity and specificity of the current technology is very low, and the results were not very consistent. So it has been more successful, people working in identification of proteins, and people as us working with the analysis of the chromosomal contents of the embryo. DNA analysis and even RNA analysis has been much more successful than metabolomics, because the sensibility and the specificity of the technology for this type of analysis is bigger.
Thanks again, Dr. Carmen. This is Dr. Angel Van. She has a question. How do we analyze the data of SBM? Do we have a special algorithm for this?
Yes, yes. As I mentioned, we had from our last publication, this gave us the basic to develop an internal algorithm with our bioinformatician team. So we have the results of the trophectoderm per chromosome, and we have the results in the media, like the sequencing result, the rough data. And we have been searching for each chromosome what should be the threshold to have the highest concordance with the trophectomy biopsy. So we have been working per chromosome, trying to weigh a little higher the sensitivity rather than specificity in the media. And then we have been these scores to each chromosome, and then the combination of the results in the embryo, we are giving the result also for the embryo. So we have been working with this. And I would like to add that now we are working with version 1 of the algorithm because we have the results of the trophectoderm in these more than 1,000 embryos. This study is ongoing. We are going to have more information about the inner cell mass. So at the end of the study, we would like to have information from at least 300, 500 inner cell mass. So the algorithm will incorporate in maybe 1 year the analysis also of the inner cell mass. And also we are going to do the clinical follow-up until delivery of the cases included. So at the end, the idea is that the test will predict us with embryos to prioritize first to have the goal of a baby at home. So this is the way in which the algorithm is going to be evolving in the next year. So we are working now in the second version. So we are very happy with the results of version 1, and this is what we are going to apply for our clinical studies and also for the clinical translation. But for sure, we keep on doing research, and we are working in the next part, in the next second algorithm.
Thank you very much for this elaborate answer, Dr. Carmen. Again, this is Dr. Rita from Bangalore, who had an earlier question. Her second question is, since older women tend to have few blastocysts due to poor ovarian reserve and other things, false negative results can be disastrous. Overall, the idea of euploid testing in them is really not convincing. What's your thought?
Well, I think that everything with a new technology is based in learning how to understand the technology and the concept. So we know that we are with a very new technology offering new information, and we need to learn how to explain this information to patients and also how to get the benefits from this information for the care of our patients. So if I am a patient of 40 years of age, I have 2-3 embryos, and I'm doing the analysis of the media, or I'm doing an IVF treatment. If I'm not testing my embryos, I'm just transferring the embryos without knowing the risk. I know I'm 40 years old, then I have a risk of chromosomal abnormalities, about 75% in general. So I have 2, 3, 4 embryos, and in general, for each embryo, the probability of being chromosomally abnormal is about 75%. If I apply the analysis of the media, I can have a more individual aneuploidy risk for each embryo. So we can have an assessment. If I have 3 embryos, maybe one embryo has the probability of being aneuploid of 20%, the other one 30, the other one 60. So it will allow me to be able to minimize the risk of transferring an aneuploid embryo. And I think this is very useful information for me. I know because I'm old that maybe I don't have any embryo, any media that can be 100% certain that is euploid, but I want to get pregnant. This is my last opportunity to be a mother. I don't want to discard embryos. So if I'm going to have a transfer, let's try to have the transfer of the embryos with better prognosis. And also it's going to give me additional information about the probability or the recommendation of undergoing a prenatal testing, because it's important to be aware of the risk of when we are transferring embryos and we have advanced maternal age. So I think this is giving us individual information about the risk of each embryo instead of the general information of the risk we have according to age. So I think it can be really of a great benefit. We are always talking about personalized medicine. I think this is personalized information for each embryo and not just a global estimation for a patient.
And that too without any trauma to the embryo, if we are talking about embrace, without any harm to the embryo, without doing any biopsy, without,
exactly.
Manipulating the embryo and all.
Exactly.
Well, Dr. Beenal is there. Here's a question. Is this useful to detect, is Embrace useful to detect translocations in genes?
Okay, thank you very much for the question. At this point, we don't advise to offer this to patients with a translocation in the karyotype or to patients with a monogenic disease like cystic fibrosis. We offer now this as a screening, so as a biomarker with information for couples without an inherited risk. We have done some research, you can imagine, trying to check for the concordance on those cases, and the concordance is good because we can detect small segmental abnormalities. But the point is that in patients with an inherited situation, we need to use really more accurate technology. So for patients with inherited conditions, we think that the trophectomy biopsy nowadays is still the gold standard technology. Maybe it changed in 1 or 2 years, but at this point that we are at the beginning of this non-invasive era, we consider that to use this for patients that without a known genetic condition.
Thanks, Dr. Carmen. I hope this addresses the question of Dr. Beenal. We have a question from Dr. Fasina. She is from the state of Kerala, India. Can we use the spent media from poor quality embryos to send for the analysis? Or, in other words, in your experience, is the morphology of embryo important in this non-invasive technology?
Okay, thank you very much for this question. In our big study, we did a multivariate analysis to see the impact of the quality of the inner cell mass and the trophectoderm, and we didn't observe any association. And also we had similar results in the embryos were starting to hatch or not. But the truth is that all the embryos had a minimal quality because all the embryos were candidates for biopsy. So we didn't observe differences in the impact of morphology if we were considering embryos for biopsy, but there are other groups working in this, and there is a Monash Institute in Australia is applying this technology for patients exactly with poor morphology. So they are applying to those cycles or those embryos that the morphology is so-so, is not very good, and they don't feel comfortable going through the biopsy because sometimes, mostly when we have a linear trophectoderm, we know that the biopsy is difficult and sometimes is non-informative. So they are offering this for these embryos with a poor morphology. So I think that as far as we have a blastocyst with a clear cavity and a blastocyst with inner cell mass clearly identified, we can apply it despite of the quality of the trophectoderm.
Thanks, Dr. Carmen. I hope, Dr. Fasina, it could address your query, this reply. Dr. Carmen, we have this question from Dr. Sayaka Haigo, and the question reads like this. Could you please let me know, we sometimes can see some blastomere cells are coming out and are attached at ICSI hole in blastocyst stage. These blastomeres are left in the media when we wash blastocyst 5 times by pipette. do we better aspirate these cells with medium, or medium for analysis should be completely clear?
Okay, very good question. So at the very beginning, when we were starting with this protocol, we wanted to check, and we were doing a registry in which cases we observed this kind of cells that they were not compacting and in the blastocyst. So we were checking if the concordance rate and the results for those embryos with these cells, but the cells inside the zona pellucida, because I want to emphasize here that as we are not doing assisted hatching before, when we are testing the media, the embryos had intact zona pellucida. So we didn't have the possibility to have cells secreted to the media. So we are sure that we are analyzing DNA that is not from cells, because the cells were still inside the zona pellucida. And also, previous to these non-invasive approaches, several years ago, we were doing the biopsy in embryos donated for research of these isolated cells in the blastocyst, and trying to compare the results of this cell with the trophectomy biopsy. And the results were very discordant. So some people suggested in the past that maybe we can biopsy these cells, but the truth is that they are not really consistent. So our idea is that these cells are not contributing to the real assessment of the chromosomal status of the embryo. And in the media, that in the protocol that we are using, these cells are still inside the zona. They are not secreted to the media, so we are sure we are not analyzing these cells. I don't know about other studies in which they are making the assisted hatching and the whole, but in our studies, these cells remains inside the zona.
Thank you very much again, Dr. Carmen. Now we have this question from Dr. Hema. She's from the city of Hyderabad in India. Her question is, would you recommend Embrace as an alternate to PGT-A?
Asa, can you repeat the question, Vikram, please?
Yeah, sure. Dr. Hema is asking, would you recommend Embrace as an alternate to current invasive PGT-A?
Okay, I'm not going to be very strong in this question, because I think that at the moment we are now with an average of 85 concordant rate of the media with the trophectomy biopsy or with the inner cell mass, I think that still trophectomy biopsy is the most accurate way of diagnosing an abnormality in the embryo. So trophectomy biopsy has a predictive value of about 95% of the chromosomal status of the embryo, and with the non-invasive, we have a predictive value of about 85%. So still trophectomy biopsy is better. But again, if we think in the comparison with the prenatal testing, it's the same thing that amniocentesis. Amniocentesis is much more accurate than the image analysis or non-invasive analysis with cell-free DNA. But again, if we can avoid the invasive approach, many couples are going to choose for a technology that is non-invasive and have still a very good predictive value. So I think we have to balance between these. So non-invasive is safer, it's not having an intervention in the embryo, no biopsy. The predictive value is very good, but the invasive technique has a higher predictive value. So we need to balance these two questions with the couple and with the clinicians to find the best choice for the couple according to their previous reproductive background, and also taking into consideration the emotional stress of the couple during the treatment. So some couples are more stressed about the potential damage to the embryo. Other couples are more stressed about the risk of having a baby with a chromosomal abnormality or having an interruption of pregnancy. So we need to balance these different emotions in the couples that we have in our centers.
Thanks again, Dr. Carmen. We have again one more question from Dr. Rita from the city of Bangalore. She wishes that the concordance between trophectoderm biopsies and spent blastocyst media could have been better. in order to encourage her to replace the current module of doing PGT-A with Embrace. So what's your say on that?
Yeah, we also would wish to have concordance rate of the media with the inner cell mass of 95%. This just would be perfect. The truth is that we have to start by the basics, is that we can never have a 100% concordance rate because we know there is mosaicism. I think that we know that the incidence of mosaicism as a biological phenomenon in the blastocyst is going to be something between 5 to maybe 8%. So this is going to be a limitation either for the trophectomy biopsy and for the media, because mosaicism is a real phenomenon. We think that thinking in mosaicism, the media can be even better than the trophectoderm, because the media, we think that is having DNA from many more cells. And the thing that we have to work strongly is into minimize the risk of contamination. So we know that we can decrease this risk, and we really are working hard to increase the concordance rate up to 90–92%. We have some centers in one of our studies at the very beginning that has this 90% concordance rate. So we think this is something that we can achieve if we work very close to the labs. But it's never going to be 100% because the mosaicism is going to be always there. So I think the highest we can go, either with trophectomy biopsy and with the media, is going to be something between 90–95%, never 100%.
Thanks again, Dr. Carmen. We have Dr. Nelson Vincent, who is extending his thanks to you for this lovely presentation today. And he has a question also. The question reads like this: was there a relationship between the volume of the spent culture media obtained and the results of DNA analysis?
Okay, yes, I will say yes. And we experienced it at the very beginning that with a big volume, we need to make aliquots of the volume to do the NGS experiment. And this additional step that we did in our lab, that we collect all the medium and we cannot analyze all the volume together because of the limitations of the protocol, we needed to do aliquots and to analyze separately and then to put together. And this was a very big limitation at the beginning. That's why we decided to decrease the drop. And several authors also went in the same direction, growing the embryos in 5 microliter drops. For us, we thought that 5 microliters was too small because the embryologists maybe would not feel comfortable, and 10 microliters, 10 to 12 microliters in some labs is the regular practice, and it's still okay. So we started with these 10 microliters, 10 to 12 microliters, and we think that for regular culture conditions, this is very consistent, this is very good. And that means that we can aspirate the volume and we can analyze all the volume in the same experiment in a single tube. We have modified our protocol to allow the proper volume of reagents, and everything, all the reaction can be performed in a single tube, and it has increased the efficiency of the test. But I have to add also that because embryoscope is in some countries, many couples have the embryoscope, and the embryoscope wells needs higher volume, we are doing some improvements in the analysis of media from the embryoscope, and if the centers can culture the embryos in 20 microliters and we can retrieve 15 microliters, the results now are encouraging. So the volume has been very important for us for regular culture conditions, but we can really improve the protocol, and even with 15 microliters, this is going to work.
Thanks, Dr. Carmen. I think the time is running fast and we are about to conclude, but I wish if you can quickly address these 2 last questions. One is from Dr. Vijay, Bombay, city of Bombay, India. Well, his question is, what is the minimum volume required to perform cell-free DNA test?
Okay, so as I will say, we will ask you to grow the embryos in 10 microliter drops, And then you will need to remove the embryo from the drop with the minimal amount of media, and ideally a volume of about 7 to 8 microliters should remain in the drop. So this will be the ideal volume. So if you have to remove the embryos with maybe 1 or 2 microliters to transfer to the vitrification dish, and then ideally we would like to collect something between 7 to 8 microliters from if the original volume is 10. The idea is to try to collect most of the volume in which the embryo has been growing.
Thanks again. And Dr. Carmen, this would be the last question. This is from Dr. Varsha from the city of Bangalore. Will cell-free DNA analysis be helpful in mosaic embryos?
Okay, I don't know. I think at this point we don't know if we have an embryo that is originally mosaic, if the analysis of the sperm blastocyst media is going to be more accurate or not. We don't know, because we have few cases in which we have mosaicism in the trophectomy biopsy and we test for the media. I can tell you that in most of the cases, it were very few in which the trophectomy biopsy was mosaic. When we were analyzing the media showed the full aneuploidy. So the media was giving us a strong evidence that the abnormality was real. So this was the only message I can give you based in very few mosaic embryos, because at Igenomix, our incidence of mosaicism is very low. That's why I didn't have a big approach. We think that maybe the media is giving us more comprehensive information for many more cells. So we think that in the future, maybe it's going to be very robust.
Thank you very much, Dr. Carmen, for addressing these questions. And I'm sure the audience has accepted the replies and they have understood the concept of embrace. And it was wonderful, Dr. Carmen, to have you here with us today. There are many doctors in the audience who are extending their gratitude to you for this lovely presentation today. I would like to name them: Dr. Hima, Dr. Rita, Dr. Chirag, Dr. Anusha. They are extending their thanks to you for today's presentation. Thank you very much, Dr. Carmen, for sparing your time. And before I wrap up, I would like to thank this wonderful audience. It was really an interactive session. We got a lot of questions, and I regret if we have failed to attend some of the questions because of the time constraints. But rest assured, you will be receiving a certified mail from Igenomix headquarters foundation tomorrow or day after. It was wonderful, everybody in the audience, to have you here with us. Thank you very much. But before I wrap it off, please don't forget to sign in for the upcoming episode of this webinar series, which is held under the aegis of Igenomix Foundation. Thank you very much. Have a good evening. Thank you, Dr. Carmen, once again.
Thank you very much, and thank you for your interest and the fruitful discussion we have had today. Thank you.
Thank you. Thank you.